Hello and welcome to our discussion today, and we will talk about a very important topic, and that is the activation of the leukocyte and what leads to the activation of that leukocyte. And once the leukocyte has been activated, what are the structural and functional outcomes that happen to mediate and facilitate the role of these phagocytes to be able to engulf necrotic cellular debris and to engulf these invading microbes, whether they are bacteria or viruses or fungi or a parasite and so on. So now we are at the site of inflammation. So we already have an inflammation, and we are in fact in the peak of the inflammation. We are at the site of the combat between the phagocytes, between these leukocytes that are able to phagocytose these necrotic debris and the microbes. And now let's talk about what we will discuss today, the objective of our discussion for today. The objective of our discussion today will be the activation of the leukocyte. We have these leukocytes circulating our blood, circulation, our blood vessels, and they are inactive. It makes sense that these leukocytes are inactive because the energy expenditure that comes with having a circulating leukocytes will be very expensive. So we need to have them circulating in an inactive form. Once they get to the site of the inflammation, then they will be activated where they can perform these energy dependent and enzyme dependent defensive functions of engulfing these microbes and properly digesting them and getting rid of them in a way that does not harm the surrounding. Okay, so a brief review to see where we at exactly in our chain and sequence of events in inflammation. We said we have a normal tissue and we have normal cells. These normal cells endure stressful or noxious insults, and that stressful insult could be ultraviolet radiation, it could be an infection by any form of microbe, bacteria, virus, fungi, parasite. We can have a chemical stress such as some detergent or chemical substance that causes harm or corrosion. We can have physical stress such as very high temperature or very low temperature, and these, or maybe a trauma like a contusion or a bruise, and the cell is equipped with built-in systems to overcome that stress. However, if the stress is very severe and the duration of it is very long, we will now have an injured cell. That injured cell will move on to the stage of being an irreversibly injured cell. We have a reversible injury and an irreversible injury. And then if we moved on to having a damaged cell in irreversible injury, then the cell will move on to cell death. And obviously the cell will not die by apoptosis, it will die by necrosis. And if the cell dies by necrosis, we will move on to inflammation. That is the chain of events that leads to inflammation. Cellular stress, very severe and long in duration. Cellular stress that leads us to have an irreversibly injured cells. These irreversibly injured cells will die by necrosis, and then after necrosis we'll move on to acute inflammation, and that's where we are. In acute inflammation, right after the damage takes place, we have resident sentinel cells like police precincts that detect fragments of this necrotic debris and detect, lipopolysaccharides or glycoproteins that have been consistently in the surface of invading cells, microbes, and once these sentinel resident cells, and we talked about these cells, they are the mast cells, dendritic cells, and macrophages. These cells, once these resident sentinel cells like police precincts, once they know and detect these foreign invaders or necrotic cellular debris, they would call and contact circulating blood, circulating white blood cells in our blood vessels, and they don't contact by phone, but they release mediators, and these mediators like histamine and like cytokines and chemokines and leukotrienes and interferons and chemokines, they cause vascular changes in the blood. And what happens is there will be endothelial gaps because the endothelial cells will retract, and these cells will start to recruit and grab circulating white blood cells, and the white blood cells will actually leak through these endothelial gaps, and they will pave the path for themselves using proteases that degrade the extracellular matrix in order to pave their way and follow down chemical gradient, these chemokines that lead them to the site of the inflammation. So now we have arrived to the site of the inflammation. So let's look what we have here. Okay, once we arrive to the site of the inflammation, we have three events. We have three stages. The first stage is extracellularly, outside of the white blood cell, outside of this immune cell that is coming to defend the body. And the two steps that follow next, or the two stages, happen intercellularly. Extracellularly, we have the recognition, the process, where's my laser? We have the process of the recognition of microbes and mediators. So as we said, these phagocytes, they will travel down concentration gradient, and they will detect N-formylmethionine. N-formylmethionine is known standard substance that is found on the bacteria. And there are also chemokines that are sent by resident sentinel cells in the damaged tissue. And there are also lipid mediators that are sent, and all of these signals are detected by G protein coupled receptor. So that's the first step, the recognition of the microbial or the bacterial N-formylmethionine, or the recognition of chemokines that cause cytoskeletal changes, leading to actin and myosin filaments to protrude towards the site of chemical gradient and move down chemical concentration. And there are also lipid mediators that cause, such as prostaglandin, that cause the phagocyte to move towards the site of the inflammation. This is what we have here. What do we have here? This is our, right here, this is the cell membrane of the phagocyte. This is the cell membrane of the phagocyte, and that is the first form of receptor that we have, and that is the G protein coupled receptor. It detects N-formylmethionine and chemokines, lipid mediators. That's the first step. It's the recognition of these signals. So that leads to stage number 2, and that is the cellular response. In the cellular response, what we have is cytoskeletal changes and signal transduction that leads to chemotaxis, which is the actual movement of the phagocyte towards the site of the inflammation following these chemokines. And there will also be increased integrin avidity, where in the endothelium we have these leukocytes adhering tightly and stabilizing in the endothelium in order for them to be able to be dragged from the blood circulation so they can leak out and move through the interendothelial gaps and move to the site of the inflammation. That's the second stage, cellular response. First stage was the actual detection of all these signals, these pro-inflammatory signals, and then the second stage will be the cellular response. In this case, it's the cytoskeletal changes and signal transduction and increased integrin avidity and chemotaxis. Then the last step will be the functional outcome. Once this G protein linked receptor has detected all these signals, these pro-inflammatory signals, the N-formyl methionine, the chemokines, and the lipid mediators, what happens is, as we said, the second stage will be the cellular response, where there will be cytoskeletal rearrangement and signal transduction that culminates in increased integrin avidity and chemotaxis. The third stage will be the functional outcome, where the phagocyte will perform adhesion to the endothelium, and there will be migration into the tissue. So now this receptor manifests its functional outcome as the actual leakage and the movement of this phagocyte out of the blood through the interendothelial gaps, when the endothelial cells retract and open these spaces through which the leukocyte will migrate. We have another type of receptor, and that is the toll-like receptor, or the TLC. In the toll-like receptor, what we have is in our toll-like receptor, what we have is there will be a detection of the lipopolysaccharides that are on the surface of the microbes. And once the toll-like receptor detects these microbial substances, such as the lipopolysaccharides on the surface of these microbes, there will be an intercept. So that's the first step. That's the recognition of the microbe or the mediator. There is a recognition of this lipopolysaccharide by the toll-like receptor, and that will lead to an intercellular response that culminates in the production of mediators such as arachidonic acid metabolites and other cytokines, and that moves on to the third stage, and that is the functional outcome, which is the amplification of the inflammatory reaction. The third type of receptor, we have the cytokine receptor that binds interferon, gamma, for example, and interleukines, and that leads to the production intracellularly of cellular response manifested by production of reactive oxidative species and lysosomal enzymes. And these lysosomal enzymes damage and destroy microbial and cause microbial activity of leukocytes that leads to the killing of the microbes. There's another, our last receptor will be the phagocyte receptor. This phagocyte receptor will also, just like the toll-like receptor, it will bind the lipopolysaccharides that are found on the surface of the microbes, and that leads to intercellular response. This intracellular response causes phagocytosis of microbes into the phagosome that culminates in the microbial activity of leukocytes and killing the microbes. This is a very important diagram, and as you see, I have divided it into three stages. One stage outside of the membrane of the phagocyte, and that is the recognition of microbes and mediators by these different types of receptors: G protein linked receptor, toll-like receptor, cytokine receptor, and phagocytic receptor. The second stage will be a cellular response that is unique to each receptor. And then in the end, after this orange line, we have a functional outcome. So we have our recognition of the microbe and the mediator. That's the first step, extracellularly. Intercellularly, we have a cellular response, and the last step is the functional outcome that is unique to each receptor. I would like to go over this step one more time with you, but we will go over it, but let's just, while we're at it, let us just look at what we have here. We have, this is the phagocyte, this is the leukocyte. The leukocyte binds it has receptors, as we see here. These receptors, they bind the microbe and they engulf the microbe. So this vacuole right here, it's called the phagocytic vacuole. The phagocytic vacuole does not have a microbe, or partially has a microbe. When the phagocytic vacuole is able to engulf this entire microbe, we will now have a phagocytic vacuole. This phagocytic vacuole will undergo another reaction, and that is to bind a lysosomal vacuole. So we have two vacuoles that will bind together. A vacuole that possesses intercellular lysosomal enzymes, and a vacuole that possesses a microbe. See, now they join. When our phagocytic vacuole joins the lysosomal vacuole, we will have this one big vacuole called phagolysosome, or phagolysosomal vacuole. It contains our microbe and it contains our lysosomal vacuoles, our lysosomal enzymes. And then the lysosomal enzymes will degrade that microbe inside the phagolysosome, and then it will properly get rid of it. This reaction right here, it shows you in detail what happens inside. We have proteases that are inside, and we also have reactive oxidative species. Reactive oxidative species are there, and we also have nitric oxidative species. So we have nitric oxide reactive species, and we also have oxidative reactive oxidative species. So this reaction is very important because it shows us that at the cellular membrane of these intercellular vacuoles, we have a cytoplasmic oxidase that is inactive. However, when we engulf this microbe, this cytoplasmic oxidase, it becomes activated and it converts NADPH into NAD+. And that converts oxygen into superoxide. And superoxide is the first type of our reactive oxidative species, and it damages completely the microbe. Then we can convert this superoxide into peroxide by simultaneous dismutation. And what happens is we have another family or class of enzymes known as myeloperoxidases, and what they do is they join a halogen such as chlorine to the peroxide, and that gives us OCl radical. And this OCl radical is another type of reactive oxidative species that these phagolysosomes use to destroy the microbe. Another type of reactive species will be the nitric oxide, where the arginine will give us the nitric oxide, and we will have nitric oxide peroxidase. Nitric oxide peroxides that will, just like the reactive oxidative species, we have nitric oxidative species that will also damage our microbes. So our discussion for today is fairly simple. Our discussion for today is fairly simple. We talked about having this membrane. This is the membrane of the phagocyte that has been recruited to the site of the inflammation. And we have 4 different types of receptors. We have G protein linked receptor as seen here. We have toll-like receptor, and we have cytokine receptor, and we have a phagocytic receptor. The G protein linked receptor has 7 transmembrane proteins, and it binds N-formyl methionine, and we know that N-formyl methionine is found on the surface of the bacterial cells. It also binds chemokines and lipid mediators, and that leads to intercellular cascade that culminates in the recognition of the bacteria and the chemotaxis, and moving towards the site of the infective agents. And there are also lipid mediators that provoke this intracellular response once they bind to our G protein link receptor. And the second step will be the cellular response, which is manifested by increased integrin avidity and chemotaxis, and that leads to the functional outcome. The functional outcome will be adhesion to the endothelium and migration into tissues. Then we also have toll-like receptors. Toll-like receptors are unique in binding lipopolysaccharides of microbes, and then that leads to intercellular production of mediators like arachidonic acid, metabolites, and cytokines, and that leads to amplification of the inflammatory signal. Then we have cytokine receptor like interferon, gamma, and interleukins that bind these signals, interferon, gamma, and interleukins, and that leads to the production of reactive oxidative species in the lysosome and lysosomal enzymes, and that leads to microbial destruction of intracellular and the killing of intracellular engulfed material. And then last but not least, we have a phagocytic receptor that binds the lipopolysaccharides of the microbes, leading to intercellular events like phagocytosis of microbes in the phagosome, and microbial activity of the leukocyte and killing the microbes. And then we have myeloperoxidases that participate in the halogenation destructive reaction of using oxygen with chlorine to destroy these infective agents. And we have the oxygen, we have the membrane oxidase that converts oxygen into superoxide, which is the main reactive oxidative species. And then we bring down superoxide into peroxidase, and we also have arginine, from which we get nitric oxide in order to get nitric oxide reactive species to destroy our microbes. So the last thing I want to leave you with that is very important for you to understand, and that is, if we have an imbalance where we have so much reactive oxidative species, that would lead to so much damage, collateral damage to nearby tissues and nearby cells that are not affected by the inflammation. But if we have genetic defects that affect the enzymes that create for us reactive oxidative species, we will have autoimmune, or we will have we will have immunodeficiency diseases where we are unable to destroy these engulfed microbes. So I hope this makes sense, and if you have any questions, leave them down in the comment section, and thank you very much.